अस्सलाम वालेकुम। आज जो हम लोग डिस्कस करने वाले हैं वो है स्टेप्स ऑफ ग्लाइकोलाइसिस। ठीक है, ये मैंने एक पूरा फ्लो चार्ट बनाया है कि किस तरह ग्लाइकोलाइसिस के स्टेप्स जो हैं वो परफॉर्म हो रहे हैं। सिर्फ आप लोगों के लिए मैंने इतनी कलरिंग करके और इतना प्यारा मैंने वो किया है, तो प्लीज़ इस वीडियो को लाइक, एक सब्सक्राइब वगैरह और इसको शेयर करें प्लीज़। अब हम लोग स्टार्ट करते हैं अपना टॉपिक। ठीक है, फर्स्ट ऑफ ऑल वी हैव अ स्ट्रक्चर ऑफ ग्लूकोज। ठीक है, इसमें आप देख सकते हैं ये रिंग स्ट्रक्चर है और ऊपर जितने ग्रुप्स हैं मैंने उनसे डिफरेंट कलर से आपके लिए लिखा है। अच्छा, इसके बाद हमारे पास क्या होगा कि एक एटीपी खर्च होगी, मीन 1 एटीपी इज़ कन्वर्टेड इनटू द 1 एटीपी and the enzyme which are involved in this reaction are two. First of all is hexokinase enzyme, which could be used, and the second enzyme is glucokinase, which can be called as hexokinase 4. Okay, now due to these enzymes this glucose is converted into the glucose 6 phosphate. ठीक है, now if we want for to this reaction to reverse, then we use the enzyme which called as glucose 6 phosphatase. Glucose 6 phosphatase enzyme cause glucose 6 phosphate to convert it into the glucose. Now we see that the hexokinase enzyme can cause other sugars, for example glycogen can be converted into the glucose 6 phosphate in the presence of hexokinase. But if we have enzyme glucokinase, then we could only convert glucose into the glucose 6 phosphate. So therefore if we have hexokinase, then we could convert glycogen, which is called can be called as glucose 1 phosphate into glucose 6 phosphate. By this, that it has one phosphate group on the first carbon atom. This phosphate is removed and then it converted it into the glucose 6 phosphate. Now this glucose 6 phosphate is converted into the fructose 6 phosphate by an enzyme which can be called as fructoglucoisomerase. हमेशा always isomerase enzyme is used to convert glucose 6 phosphate into the fructose 6 phosphate. This isomerase enzyme causes the interconversion of glucose into the fructose. Now we have 1 phosphate attached at the 6th carbon atom while the whole structure of fructose remains same. Now another ATP is used. This ATP is converted into the ADP and the enzyme is phosphofructokinase 1. And the phosphofructokinase 2 can also be used, but phosphofructokinase 2 convert phospho 6 phosphate into the phospho 2 6 biphosphate. But if we use phosphofructokinase, then it converted into the phospho 1 6 biphosphate. Now for the reversible of this reaction, we use the enzyme which called as hexobiphosphatase. ठीक है, hexobiphosphatase enzyme convert 1 6 fructose 1 6 biphosphate into the fructose 6 phosphate. Now we see that due to the conversion of ATP into the ADP, one more phosphate group is attached with the carbon number 1. So therefore one phosphate is at carbon number 1 and the other phosphate is at carbon number 6. Now this phospho 1 6 biphosphate converted into the glyceraldehyde 3 phosphate, mean G3P, and the dihydroxyacetone phosphate. ठीक है, this is the structure of glyceraldehyde 3 phosphate. It has phosphate group at the last 3 carbon, and the dihydroxyacetone also has a phosphate group at the last carbon atom. Now this hydroxyacetone converted into the glyceraldehyde 3 phosphate because all these are isomers of each other. Now this glyceraldehyde 3 phosphate converted into the 1,3 biphosphoglyceric acid. This is the formula of 1,3-glycerophosphoric acid. It has phosphate group on the carbon number 3 and the carbon number 1. So it is biphospho 1,3 and the glyceric acid mean that it has this group. So therefore it is called a glyceric acid. Now it is converted, 1 ATP is converted into the 1 DP is converted into the 1 ATP by the enzyme triose phosphodehydrogenase, and the 1 ATP is converted to NADH. This H comes from this phosphate group is removed and an H attach with it. Okay, so due to this triphosphate dehydrogenase is used in this process. Now an ATP is converted into the ADP is converted into the ATP, and the phosphoglycerokinase enzyme is used, and this is converted into the 3-phosphoglyceric acid. Mean one hydrogen is attached with this oxygen, and this phosphate group is removed, and this phosphate group is attached with the ATP, and this structure is called as 3-phosphoglyceric acid. Now we use enzyme which is called as phosphoglyceromutases. This enzyme converted into the 2-phosphoglyceric acid, mean that this phosphate which is attached at the third carbon atom would replace into the second carbon atom, and the 2-phosphoglyceric acid is formed. Now this 2-phosphoglyceric acid by the enzyme enolase and by the removal of water, it converted into the enol pyruvate. Enol pyruvate has the formula same, but the difference is in this group. This group has 2 carbon atom, but this OH group is removed to form water, and double bonded CH2 is formed, which is called as enol pyruvate. Now we use enzyme which is called pyruvate kinase, and 1 ATP is formed, and then pyruvic acid is formed, which formula is COOH, then C double bond O and CH3. So in this way the glucose is converted into the pyruvic acid.